Hello, everyone. I'm Train Dahlin, PacBio's Director of Emerging Applications, and I want to welcome everyone to the PacBio webinar series with today's topic focused on understanding the challenges of AAV sequencing and how experts in the field of gene therapy are using HiFi reads for better and more efficient vector design and quality control. So as you know, gene therapy and cell therapies are now viable medicines that have reached the market and providing transformative benefits, meeting large unmet needs. And although the development of vector-based gene therapy is accelerating at a rapid pace, there are still many challenges that still need to be solved to facilitate the development, design, and manufacturing of these vectors. So today I'm joined by three great researchers in the field of AAV vector gene therapy who are going to share their insights and current practices for developing and validating AAV vectors. Our speakers today will be Elizabeth Tseng, who is the Associate Director of Product Marketing at PacBio, followed by Don Selby, who is the Principal Scientist from Homology Medicines Incorporated, and then finally Terrence Hanscom, also a scientist from Homology Medicines Incorporated. So we have a lot of great material to cover today, and the presentation portion of the webinar will be followed by a questions and answer session. You are welcome at any time during the webinar to submit your questions by typing them in the area provided on your attendee control panel, and we'll get them at the end. We've also uploaded a number of complementary pieces of literature to your control panel, so please feel free to download them and take them with you. And finally, we will be recording this webinar and making it available for download in the next few days. So please keep an eye out for a follow-up email with a link to the recording. So with that, let's get started. So our first speaker for today will be Liz Tseng. Liz is currently an Associate Director responsible for IsoSeq and emerging applications in the Product Marketing Department at PacBio. So welcome, Liz.
Hi there, can you hear me well?
Yes.
All right, so let's get started. And first of all, I apologize. There's active construction going on next to me. I did let the other organizers know, should there be disruptions or a break in my presentation, Tran will take over and we will proceed the webinar with the remaining speakers, and I will try to get back on as soon as I can. So today I will start the presentation by talking about generally what HiFi reads are and how you can use them for AAV sequencing. So let's start with what smart sequencing is, how people have been using it. We can sequence anything from viruses to eukaryotes to bacteria. All you need to do is isolate the DNA and RNA. And I mentioned this is because PacBio has been used not for just AAV vector sequencing, which Don, one of our guest speakers, will talk about, but will also be used for sequencing RNA for the looking at the transcripts that's being expressed later in the talk today. And people have also used smart sequencing for sequencing the plasmids. The way you can get sequencing done is once you have isolated the DNA or RNA, create a SMRTbell library by ligating the adapters onto double-stranded DNA. Sequencing is done on our Sequel 2 or 2E systems, where the sequencing unit is called a SMRT cell that contains millions, or currently 8 millions, of what we call zero-mole waveguides. And what happens is once these molecules are loaded into these ZMWs or zero-mole waveguides, sequencing is happening in real time as DNA polymerase goes around this template and incorporates nucleotides anew. And once that happens, there is fluorescent signal. The fluorescent signal is detected by our machines and able to call the bases. What I'm showing you here is that because it's happening in real time, there is no amplification. Nucleotide incorporation includes kinetic information. So you'll see later in Don's talk that you can actually use this kinetic information for your vector design. I will explain the next slide how the data is presented, what is actually in HiFi read. So HiFi reads are based on what we call circular consensus sequencing. Looking at the structure, the SMRTbell, once the SMRTbell adapters are ligated on, it actually forms a double-stranded structure. And then you're actually, when sequencing happens, the DNA polymerase goes around both strands, actually now in a circular fashion. It will sequence the insert both for the gold and the purple portion. That means the polymerase can go around this circular structure many, many times. However, I know what you guys want for data-wise is just the insert. So we will actually bioinformatically call a consensus of sequencing around these different passes of the same molecule to create a consensus read that's called HiFi reads. for up to 25 kilobases of insert, we can achieve 99.9% accuracy. However, as we know for AAV vectors, for cDNA transcripts, usually we won't get to 25 kilobases of insert, so our accuracy will actually be even higher. Just to show that HiFi reads are accurate and long using whole genome sequencing data, this is a comparison of PacBio against other sequencing technologies to show that indeed we offer the highest accuracy. Sequencing has been used on PacBio to not only for AAV vector, in fact, our most popular applications are sequencing whole genome. We've recently helped complete the human genome 20 years later to fill in the 8% of remaining gaps. PacBio sequencing has been used to explain human diseases, both by sequencing the DNA and the RNA. Finally, in today's talk, you'll hear about how sequencing is used to accelerate gene therapy research. So now I would like to walk you through some of the AAV publication highlights. PacBio sequencing can be used throughout different phases for AAV. I will walk you through publications that cover discovery, such as using PacBio sequencing to discover novel AAV species. I'll cover some of the applications that covers vector sequencing and the validation once it's being produced, but Dawn and Terrence's talk will also cover more portions of these. So in this first publication that I'm showing you, a novel AAV species is discovered with HiFi sequencing. A single clinical sample was extracted using primers designed against the conserved regions of the known AAV species. The researchers were able to find an abundant new species that represented 45% of the AAV reads that were found. This new species, named AAVV66, boasted strong packaging performance and, despite being phylogenetically similar, is functionally distinct from AAV2. In the second publication, the researchers used HiFi sequencing to sequence viral particles to detect undesired backbones. In this case, they were sequencing a self-complementary AAV. Shown here in this IGV visualization, what you're seeing is the structure of this plasmid. The blue thick line covers the ITR to ITR region. If the packaging was clean and only consisting of what's between the ITRs, the reads, which are these gray lines that you're showing, plus the little rainbow colors, should only cover the blue region. However, as you can see here, the sequencing actually shows that there are detection of undesired black plasma material. Furthermore, they can also characterize that these reads are also mapping partially to the human genome, showing that there's chimeric integration. In the follow-up study, they used PacBio sequencing to look at whether the incorporation of single-guide RNAs would actually result in undesired truncation. What they found was that sequencing revealed that the vectors would only cause truncation if it's carrying a dual single-guide RNA expression in a tail-to-tail configuration. Also, similarly to what was found in the last article, they also found recombination events between HG38, the human host, and the vector genome. Now, this study was explicit looking at integration events into human hepatocytes. Using a mouse model, they found that chromosomal integrations happened at a frequency of 1 to 3%, both in vitro and in vivo. They were also able to characterize what were the most common insertion elements. And perhaps not so surprisingly, they were elements that incorporated the ITR region. This latest publication explicitly used HiFi sequencing to look at production differences between HEC293 and SF9, two of the most popular production platforms. What they found first was when looking at the gel, they were able, which is shown here on the upper left-hand corner, they were able to see that there is a difference in the banding of the sizes. It was expected to be around 3.3 kilobases. The HEC293 band seemed to be cleaner. The SF9 band seemed to be have smear. They first did sequencing using short Illumina short reads, But looking at the Illumina short reads, which is in the C panel of the upper left-hand corner, they weren't able to actually see any differences using short reads between the HEC293 and the SF9 dataset. They hypothesized this was because the strong secondary structures of the ITRs and the GC content made it really difficult for short reads to completely cover this region and show any differences. Now, PacBio being long and accurate, we could actually sequence ITR to ITR. And what they're able to show in this visualization here, where the left-hand side is the visualization of the reads aligned to the plasmid using that was produced by the HEC293, and on the right-hand side are the reads that were produced by the SF9 platform. They were actually not able to see that there were more truncated genomes in the SF9 produced vectors. What they also found was that, which is not shown in here, was that the what was previously thought to be empty capsids actually contained ITR-bearing short DNA fragments. Finally, regardless of the production platform, they showed that ITR heterogeneity directly influences the genome population of the AAV species that's being produced. So now that I've walked you through the publications, I'd like to give you a high-level overview of the library protocol on the bioinformatics analysis that PacBio currently offers. I would like to emphasize that because AAV is such a unique vector, there's really not one way to produce a library from which you can use PacBio for sequencing, and there is not a one way to do bioinformatics analysis. What we are offering here is a guideline, and we're happy to work with any of you to understand more how we can actually help you make a better AAV library and do better analysis. So on the high level, this is what the AAV workflow looks like. We have a published protocol, which you can find on our website, that offers a way to prepare AAV vectors into a SmartBio library. With our latest SmartLink, so our software is called SmartLink, with our latest SmartLink release, you can actually run something called AAV mode on the instrument, which will inherently understand the differences between self-complementary AAV and single-strand AAV. And finally, I will show you that we also have a command line bioinformatics tutorial that will also produce meaningful plots that you can interpret. This is what the workflow looks like. We begin with AAV DNA extraction and QC. Now, with the single-strand AAV, recall that in order to make a SmartBio library, you actually need double-stranded DNA. So for single-strand AAV, one of the methods that we're recommending in this protocol is to use thermoannealing, but as many of you might be familiar, there are other ways to create that second strand. And in fact, Don's going to talk about one of the other ways to create that second strand. Once the reads are produced, they can be mapped back to the reference genome. And then this current command line option, which I'm showing the URL down here, will produce meaningful plots such as, where's the most common map start and end position. Now for this data set, which by the way, this data set is publicly available, you can find it by accessing the link below. It shows that most of the reads actually mapped beginning and ending exactly where the ITRs are. And this is because this is a commercially purchased, relatively clean AAV vector. We can also characterize how much of the single-strand AAV is full, that is covering the ITR to ITR versus partial. And finally, I want to actually mention something that is, I think, on the horizon, but isn't actually something quite people have used PacBio for on AAV yet, is that we know that unmethylated CpG motifs in AAV genomes are potentially an issue to think about, is it might trigger immune response. Now, with the latest SmartLink release from PacBio, we can actually now natively output 5MC information. And this does not require extra sample prep because there is no amplification. And in essence, AAV library preparation is a little bit like just a whole genome sequencing library preparation. You can actually get for free your fifth base, which is the methylation signal. So to just summarize, HiFi sequencing can be used as a better tool to characterize the vector design, understand the purity, and host integration events. And we now have a on-instrument software support. We also have a command line that can generate the reports that I just showed you.
Okay, so it is my great pleasure to introduce our next speaker. It's Don Selby from Homology Medicines Incorporated. Don is a clinically trained molecular physiologist with over a decade of work in third and second generation sequencing, and he is currently developing and designing AAV vectors to deliver novel therapeutics. Welcome, Don.
Okay, thank you. Everybody hear me? All right. So yeah, thanks for the introduction. So I am from Homology Medicines. And, like I said, we are working on developing AAV vectors for gene editing and gene therapy programs. So we've kind of come up with a means to identify where our vectors, as Liz was kind of alluding to, that there are these mystery phenomena where you will want to package something into a capsid and it does not get fully encapsulated. And you really normally won't have any idea exactly contextually where it's breaking, what's problematic, what you need to rectify, and what could be actually causing this pattern you would see. So anyway, kind of on this exposition slide, you've got kind of on the left, there's conceptually, you think you've got the uniqueness of PacBio, its sequencing, its ability to look not just at what bases are being integrated, but also you can look at the modifications which you can apply to bases. And the reason you can do that is because of PacBio's ability to look at the kinetics of the actual sequencing event. In other words, you pretty much got a microscope sitting there observing the polymerase, and you can observe its activity. On the other hand, you've got on the other side of the page is the uniqueness of the AAV's biology, right? So you've got your virus, you've got it in, you want to put it in its little capsid, and then in the upper right, you can see a good example of an alkali gel there, showing you the differing lanes. You've got a lane that's close to 5 kb, so that's indicative of your idea of that's a fully loaded capsid. Whereas you have, in your other lanes where you have these smaller fragments, and that's to visualize for everyone the idea of these partially filled capsids. And conceptually, you really don't know, again, where they're breaking and why they're breaking. And in any way, then in the lower, we've kind of got this idea of you break your capsids open, and through the miracle of hydrogen bonding, where you've got your positive and negative strands, you can marry that again with the PacBio's ability to see these modifications in your bases, and you get essentially what we've got there in the middle, which is us mapping where our strands have been breaking. And so we can now target what areas of our recombinant AAV we need to go back and redesign. And obviously, really enhances our design process. So anyway, again, this is just kind of to demonstrate the idea of how the system works. Again, you've got a camera, you're looking at a polymerase, which is represented by the little blob, and here you get these flashes of light as it's incorporating in the nucleotides, right? And then in the lower, the little darker screen at the bottom is essentially to give you the idea of this observation of the actual movement of the enzyme over time. What's going on? How's it incorporating its bases, and how's it working? So, as I alluded to on the very convoluted third slide there, we have this ability to look at modifications of bases. And the way we went about it was kind of the older the way of looking at it, which is this interpulse distance. You essentially look at the period of darkness that's occurring between your incorporation of a base and its interruption, essentially anthropomorphically thinking of the polymerase is thinking, what the heck have I just been handed? Why do I have this base doesn't look like what I'm used to? And so as it essentially tries to figure out what to incorporate, you have these increases in your interpulse distance, the darkness between one base incorporation and the next. And the other nice thing is that you aren't stuck with just using one base. You can use the whole gamut of pretty much any discovered modified base leaves its own unique signature, essentially based on the spatial arrangements of the modifications, how all of that interplays with the sequencing polymerase, and the fact that the sequencing polymerase is in contact not with just one base at any one time, but actually essentially 11 nucleotides up and downstream, and away from its catalytic component there in its binding group. So you have this unique signature for 4-methyl C, as you can see on the top frame, and then we also have the same thing with a modified 6-methyl A. And so essentially that allows us to add mixtures of modified bases to our, as we'll see in our reactions. And so it allows us to actually have a good amount of bases that allows us to identify where molecules are breaking. So again, to shift gears and go back to, what's that apply to what we're trying to fix here with our AAVs. So a bit of a review from where we're coming from on the virus. So again, they're packaged in single-stranded molecules of DNA. You get a roughly 50-50 mix of a positive and negative strand, as these things are unwound essentially in their host cells from a plasmid, because we're working with recombinant, and then we're of course stuffing them into their capsids. And the other thing is the only thing that you kind of need to bring along from the wild type AAV, obviously the capsids, obviously those proteins, but from a cis-DNA standpoint, you need, your Princess Leia hair buns there, the ITRs, are really the only thing you kind of need to worry about bringing along for the ride when you construct these things. For us in homology, we've got more flavors than this, but for what we're talking about today, we have a gene editing, which is we just go with homology arms, hence the name of our company. And we go in and we try to go through homology and try to, rectify monogenic disease. On to the right is probably a bit more canonical as far as the world's concerned. You've added a gene therapy, which is where you essentially have an episomal corrective gene that you've inserted in, and it's providing a corrective protein. So anyway, how that applies to us is, again, like I said, we go from these ideas to want to generate a therapy. So we can see here kind of on a conceptualized plasmid that I just threw together for this talk, is that you've got these plasmids, and these plasmids, again, like I said, they are peeling off your positive and negative strands. You've got ITRs that need to be in either end of that blue circles you can see in the red and the green label ITRs. So those are your hair bun pieces, which are necessary. And then those, of course, drive you getting a fully loaded capsid. Again, so the way we go about then investigating, confirming what we've got is what we've designed is actually what we're getting into our capsids, of course, is you've got then to take your capsids and to break them open. So you lyse your capsids. And now through again, like I said, the wonderful miracle that is hydrogen bonding, you will reanneal a vast majority of your population of positive and negative strands to get the appearance of what we have at the bottom with the little doggy bone there, with your ready to go into what you would say is your canonical smart bell sequencing, where you anneal your smart bells either end there at the opening and you sequence it. However, in the real world, if you were to essentially to take one of your designs that you're trying to create and test and to investigate, you're not going to have always that perfect scenario. You're going to also have this existence of partial and fully intact capsids. And so what you end up having is when you go through your process of lysing your virus capsids, you get these mixtures between some strands that are completely full, they've broken, and then they will hydrogen bond to their complement strands, but oftentimes those complement strands are not fully intact, they're partial. So with that idea in mind, you can kind of take advantage of that and bend that to your own advantage, and then interrogate, where exactly is everything breaking? So again, if we were to go back and pretend we weren't going to use modified bases, every sequencing company technology in the world, this is, just the thing. You want a shoe, you got to put it on your foot. You want to, you want to be able to sequence something, you've got to kind of do the same thing. Everybody goes through these steps of DNA damage repair. You've also got to use ligases when you do that. So you have these cocktails. And those cocktails obviously are repairing something that's say basic. They repair something that, glycosylases. It's the whole. There's only so many things we can do, and so everybody has to go through these same choke points. And this sort of stuff with library prep is one of those points. So for us, what we did is we've taken a DNA damage repair enzyme. Obviously, you've got to have some other enzymes there in your cocktail. But for us, instead of just adding your vanilla A and your vanilla C, we added 6-methyl A, 4-methyl C. And so what that allows us to essentially do is you can rebuild these patches. You can see down here on the bottom, and now we're able to actually, going through putting it on the PacBio sequencer, we can actually see where previously something had been broken. Now we can actually physically see where the repair events actually occurred during these steps of DNA damage repair, sequence them, identify them, and then of course that allows us to go back, take another, run at designing these vectors and go through the process again. And it's quite efficient and very, very, very helpful. So this is kind of an idea of the first time we were trying to get an idea of what the heck we were doing. And this is actually the results of our very first run we did with that. And we're really excited because, as you can see here, we were actually able to map out where the positive strands were breaking, where the negative strands were breaking. So, of course, that allows us to do things like you will see on the next slide, which is we can have these iterative designs. We can go back to our old drawing board and go, oh, okay, well, we need to fix this part, and this and that, and this and that. And so what you end up having at the end of the day is you have a much more efficient design process. So as far as us coming up with vectors and improving their efficiency, it's a game changer for us. One of the other things that's really kind of interesting from a biological standpoint, we also were able to detect is this funky pattern, as you can see here. So I'm going to change my pointer. Hopefully that's visible. Mm-hmm. As you can see here, you've got these particular pattern that is consistent across all of our designs, regardless of what we're doing. So this just seems to be a scientifically interesting observation as far as the biology goes of these viruses, is that there also seems to be a natural, anticipated break that's occurring at the 5' ITR. What does it mean? We don't know, but scientists, these kind of things are kind of cool and exciting. I thought worthy of pointing out. So yeah, these are really helpful tools. Obviously, this can really improve your safety profile as far as going to the regulatory agencies and all that. Saying we've confirmed that we've done the best designs that are absolutely possible with our plasmids, and they're ready to be packaged into their capsids. And yeah, it's a godsend, really. And so I'd like to thank all the folks, as you see here, at Homology, as well as the assistance we had from folks at AAV, and then obviously PacBio as well. Thank you very much.
Thank you, Don, for the presentation. And again, we'll save the questions for Don at the very end. All right, it's my pleasure to now introduce our third and final speaker for today, which is Terrence Hanscom, also from Homology Medicines Incorporated. So Terrence holds a PhD in molecular genetics with a background in DNA repair, gene editing, and short-read sequencing. And Terrence will be talking about how he uses HiFi reads to validate the expression of viral vectors to improve overall vector design. So welcome, Terrence.
That's right. Yeah. Thank you very much, Trang. Yeah, I just want to first thank the organizers, Trang and Donal, for having me here today. Can everyone see my slide here?
Yes.
All right,
Yes.
great. Thank you. All right. Hi, everyone. Yeah, again, my name is Terrence Hanscom. I'm a scientist here in R&D at Homology Medicines, and I'm very excited today to tell you a little bit about some of the work that I've been doing with targeted mRNA isoform sequencing using PacBio so that we are able to characterize our recombinant AAV therapies. So I was thinking at first it would be really nice to go into some of the background biology. So an AAV is it's a really small 20 nanometer virus with a 4.7 kb single-stranded DNA genome. And so in the wild type form, it has these two genes within it with these various different gene isoforms that are expressed. And so here we have rep and we have cap. And so rep is involved in viral replication, where cap is involved in capsid formation. And so these two genes are flanked by two ITRs at the 5' and 3' end. And so these ITRs, or inverted terminal repeats, are just these palindromic sequences. And so companies like ours are able to, just by keeping the ITRs and sort of payload in between there, and utilize these for a therapeutic delivery. And so I wanted to go into a little bit about how the AAV actually replicates. So it uses this really fascinating mode of replication called rolling hairpin replication. And so again, at the ends of the single-stranded genome, there are these palindromic sequences, which kind of self-prime. And so upon an infection, they will take advantage of the polymerases there within the nucleus of the infected cell, and it will just start replicating. And towards the end of that strand, it will just push out the other end. So basically the other palindrome, thereby making this big old hairpin right here, which then sort of collapses on itself and self-primes in the other direction. So you have this replication going back and forth until rep, one of the genes that's expressed in the wild type form, it has these various just different jobs where it can, depending on the frame in which it's expressed in and how it multimerizes, it's actually able to form a nuclease or a helicase with an ATPase domain. And so in this case, it cuts the genome at what's called the TRS site, or terminal resolution site, and covalently binds at the 5' end. And so you can see that here right up at the top, where you have a particle of rep bound at the 5' end here. And then the AAV genome is inserted 3' to 5' into the port within the capsid, and just naturally, it's able to insert about half of the genome in on its own, and then right at the opening here, it's able to utilize its helicase activity to shuttle the rest of the genome in. And then once it exits the cell and infects another cell, it's able to uncode itself in the same direction, 3' to 5'. So yeah, there. So again, we are able to utilize this AAV, sort of repurpose it, and just by putting a therapeutic payload in between these two ITRs. And so, as Dawn mentioned, I don't want to go too far into this, but just for redundancy. But we have our gene therapies, which will enter a cell, enter the nucleus, and then just kind of hang out as an episome, just expressing the mRNA, the correct mRNA and protein, and not affecting the genome, the genomic DNA. And so, this has some benefits and some drawbacks. One of the drawbacks being that the durability is is not as much as one that would integrate into the genome. So this would not replicate with subsequent cellular replications and divisions. Sometimes a gene editing therapy would be better, would better serve the disease in question. And so in this scenario, the capsid would enter the nucleus and uncoat, and then that therapeutic payload would then recombine at the locus where the mutation is, thereby fixing the mutated gene, and which creates this functional mRNA and functional protein, and delivering a healthy cell. And so the one question that's really important to us is, are we getting correct mRNA isoform expression upon delivering these therapies? Because you can potentially get a lot of differences here. Certain genes have many different isoforms, and so we want to make sure that what we're delivering to a patient is going to be what we intend. And so the question, are we getting correct isoform expression? The natural way to do that is to characterize with RNA-seq. And so this is just a real brief background of what RNA-seq is. So you have these two different samples that are represented as two different mice receiving two different therapies. You carry that experiment out, and then you isolate the RNAs. You generate your cDNA library. In this scenario, they're using the poly-A tail to generate the cDNA library. You can also use other ways, such as random hexamers or gene-specific primers. Yeah, so in this scenario, in this slide, they're using an Illumina machine, it looks like, to generate short reads, which you then, when you're demultiplexing them and bioinformatically analyzing them, you have to align them to a reference. And so this has some drawbacks, right? Especially where we're concerned, where we want to be able to understand the full length isoforms, right? And so NGS short reads don't really capture that. And this is made more apparent on this slide that I got from PacBio, actually. And so you have your gene up here. Your gene in blue is represented by these 6 exons. And so you're going to have potentially several different mRNA isoforms. And so you have some exon skipping here, you have some here and here for this isoform. So you have these 4 different isoforms that are coming from this one gene, right? And so if you're looking at, say, with short read technology, if you're looking at, say, just this read, right, you don't know if it came from this mRNA isoform or this mRNA isoform. And this is really important to us. This is central for safety and efficacy for our patients. And so PacBio's IsoSeq method, this solution is really effective at showing us, exactly what we're delivering, in terms of expression. And so what this looks like at the bench, so you carry your experiment out, you transduce, and you wait whatever time point you want to go for, and then you can extract the RNA. You do cDNA synthesis and amplification, and then you do some hybrid capture. And for those who don't know what that is, it's they are these biotinylated probes. The ones we use are approximately 120 nucleotides long, and you're just basically capturing only the sample that you want, only those cDNA sequences that you want. You can then amplify the signal after that, and then jump into PacBio library prep and sequencing, which kind of looks a lot like this. And so, yep, okay. So you have your cDNA sample. You can go through DNA damage repair. You do the end repair A-tailing, so you're putting A's on the 5' and 3' end on the 3' strand, which enables you to ligate your smart bells, which have a T on the 3' end. And so that allows for pretty efficient ligation. Then you can anneal your primer and bind your polymerase. And so you have this nice ligated circular fragment right here, which then ends up at the bottom of a ZMW. So as Liz mentioned, this is one of 8 million ZMWs on a single smart cell. And so you have this polymerase bound down there, you have integration of your fluorescent nucleotides, and you have this laser shooting up at it, and so you can see what integrates and when. And as Don was mentioning, you can really capture that signature. And so you can see over here in this animation, you see the polymerase kind of going around this circular molecule that you've created with your smart bells, and then you have your payload in here. And so you can see the polymerase displacing that newly synthesized strand, and it just goes around and around and around, creating what's called a circular consensus sequencing. And so this gives us, the confidence to know that what we're seeing is real. And so, with these multiple passes, it doesn't really matter if you have one or two mutations, because overall you get this consensus that is built on many turns of the polymerase. All right. So our goals in terms of the in-house isoseq method were to have a full picture of the expressed isoforms of our codon-optimized recombinant AAV therapy variants, to quantify each isoform if we did see any, to improve codon optimization computational models with some empirical data, and to include this as a part of our R&D pipeline when we're choosing candidates to go forward with. So I'm going to go through a little bit about how I built this assay in terms of the two different phases that we initially had with it. And so for assay development, I used a couple of in vitro samples. These were just naive primary human hepatocytes. They had not been transduced with any virus. And from there, I was looking for just a control gene, ApoE. And so ApoE is a very well characterized gene. It's involved in Alzheimer's, so it's been very highly studied, and it has this really straightforward isoform profile. It's expressed very highly in the liver, and it is not impacted by an infection of our vector. And so then I went through and I had some experimental samples. So these were in vivo samples where I used murine hepatocytes that were transduced with a test vector, one of our recombinant test vectors. And so our target gene, the one that we were hybrid capturing for, was our test vector payload. And so this slide is really just about ApoE and how it's expressed. So you can see in this heat map, this is the expression of the 5 different isoforms of ApoE, and you have all of these different tissues. And so right down here is the liver. So you can see in the liver that ApoE is expressed pretty highly. At least there are these top 3 isoforms anyway, and then there are these bottom 2 that are pretty lowly expressed. And so, we thought that if we could pick up these 2 lowly expressed isoforms, then we'd be able to, confidently say that our assay was quite sensitive, and we could be confident in looking for different isoforms of therapeutic variants that we're testing out. And so here's some of the data. And so here, what I'm doing is I am comparing an isoseq run with a whole transcriptome run of the same sample. So here we have the hybrid capture done, and this is no capture. And so you can see in the hybrid capture, ApoE, which is what we were capturing for, is a majority of the reads that we wanted, but we intended on getting. And then this is followed by HP or haptoglobin, and then albumin, and then all these other genes that are expressed in liver tissue. And then you can see in the whole transcriptome where there's no capture, you got albumin, and albumin is just blowing out the signal. It's very highly expressed, followed by all these other genes. And then you have haptoglobin as your 5th most transcribed gene, which was interesting to us. We saw this. We saw that haptoglobin in the captured sample was the 2nd most transcribed gene, or captured, and then and in the whole transcriptome it was lower transcribed, even compared to albumin. And so we found, after looking into this, that one of our capture probes actually had some sequence similarity to haptoglobin. So there is some conservation between ApoE and haptoglobin. And so, I say this because this is something to really keep in mind when you're designing your probes for a particular gene that you're trying to capture, or a particular cDNA. And it's not a problem necessarily, but you just want to be aware of any off-target capturing. And so here's another view of this. So okay, got my pointer back. All right, up at the top here, we got the ApoE locus, and we're comparing the capture versus the uncapture. And so in the captured sample, you can see that ApoE is very highly expressed in these 4 different exons. And then in the uncaptured sample, you can't really see it, but it is there. It's just, more lowly expressed. Not more lowly expressed, but more, there were just fewer reads because it was uncaptured. And then here we're looking at the albumin locus. You can see in the uncaptured sample, all these exons are very well represented across the board. And then in the captured sample, you can see a little bit of it, but what this is saying to us is our hybrid capture is a highly specific enrichment. And so here, we're also looking at some ApoE data. And so this is our ApoE data up at the top here in purple, compared to two different references. So ref 1 and 2 are from RefSeq and GenCode, respectively. And so here we are looking at the same data up at the top here in purple. It's just zoomed in. So you got a higher and higher resolution if you can appreciate the y-axis here, going from 0 to 1 million up at the top, 0 to 50,000, and 0 to 5,000. So we can focus on this 0 to 5,000 view here. And so I showed this slide to kind of show you that we were able to pick up those very lowly transcribed isoforms. And so we knew that we could be confident going into an assay with a transcribed sample, knowing that we could pick up really lowly expressed isoforms. So yeah, our assay was quite sensitive. And so this is an example of test vectors that we have sequenced using the isoseq method. And so here, up at the top, we have test vector 1, which computationally anyway had a predicted splice donor and acceptor site. That it was pretty. We were pretty confident based on one of the predictors that we were going to see some splice donor and acceptor usage. And then in test vector 2, we changed a few base pairs and tried to remove that splice donor and acceptor site. And so you can see here that we have this smaller read size here. And so what we took from this is that, and in some other data, was that approximately 10% of the time or so, we were getting some cryptic splicing. And you can see that here, that it was resulting in just a smaller mRNA. So you can see that there. And then when we changed it, we were able to see that we totally resolved that that expression of that cryptic splicing. This is another view of that. And so here we're just looking at test vector 1. So that one up top that we just showed in the previous slide. And so just so you know, transcription is going from right to left here. And so here you can see that you get your first exon, your first intron splicing out really nicely. You have your expression going across really smoothly up at the top, and then you have this cryptic splice site, right? So you can see this bump. And so this is what I was talking about. With approximately 10% of the time or so, we were actually getting this splicing, which was then resolved when we went through and changed a few base pairs. And so this was really important. This kind of strengthened some of the computational models that we'd been using, and we can use this empirical data to do that. So in summary, the isoseq method is highly sensitive, and it's able to detect rare events, including cryptic splice sites and polyadenylation signals, infrequently transcribed isoforms. It's an excellent system for isoform discovery, which is primarily what we're using it for. Also, again, be aware of sequence similarities between your target gene and other transcripts, just due to the hybrid capture. It's fine if they're there, but just be aware of it. Thorough DNase treatments are also very useful to reduce sequencing anything that you don't want to see, and then you can really strengthen your computational models with some of this data. I want to thank everybody on my team, especially Don. Don's my mentor and just great guy. Yeah, thanks everybody. Thank you, especially to Liz, who's been helping out a lot with this, and John, Jason, everybody, Samuel, and yeah, thank you all very much.
All right, thank you very much, Terrence, for a great presentation on how you're successfully using and measuring expression using PacBio. So we have a few minutes left on the call, which we'll use for Q&A. I'm looking through the panel here, and I'll see if I can find some thematics here, but maybe I can pose the first question to you, Terrence. So question is, how does, I'm sorry, excuse me. Can you tell us why quantifying isoforms are important for AAV vector design, and can this be done using IsoSeq?
Yeah, I think it can. You know, I mean, depending on the disease that you want to treat, you could theoretically engineer therapeutic vector to you know, to express, you know, one isoform or many, depending on what's appropriate. You know, you just want to be able to avoid expression of what you don't want. You know, Aziz Al-Khafaji, who was doing the mass IsoSeq, who developed that, you know, that's where I see IsoSeq really becoming quantitative, because, you know, you're essentially putting barcodes on all the single molecules. So you're able to, you know, even after all the amplification steps, you're able to see, you know, how many original molecules you had, and in that way it becomes really quantitative, which, yeah, is really important for, you know, vector design, because, you know, if you have if you're generating a therapy, and you know, you really want to know, like, how often it's you know, doing things you don't want it to, or yeah, just really just what's happening. Hopefully that answered your question.
Yeah, thanks, Terrence. This next question is for you, Don. So you talked about using long read sequencing to fully characterize and validate your AAV design. So why did you choose PacBio versus other long read methods such as ONT or even synthetic long reads for this research?
So the reasoning for doing it this method is, if you're making the toss up between this for Oxford, you can make it on two choices. One would be accuracy. So for us, going over the same read multiple times, that's obviously a benefit, as well as you have the full context. So for instance, when you come up with something with a good degree of secondary structure, which are the ITRs, those are one of the other things that, you know, you have to be cognizant of if you're going to be using a POR to sequence it through. And again, you come up with, also, you've got kind of this, it's kind of in a sweet spot in that there's a lot of tricks that you can take advantage of, like we did with modified bases regarding the PacBio, but you've also got this well-established kind of support infrastructure, as far as, you know, if I have to figure out how to go with, you know, working with slapping a smart bell on, like somebody else has also had these questions, is how do you rectify these issues with, you've got these ITRs that also need to be joined together at the end, and then you have to also put a smart bell on the end. So it's kind of where it comes in with its flexibility, and then also, I guess you'd say, reliability as well.
Thanks, Don. And maybe a question for Liz. So we have a question here, Liz. Can other viral vectors be sequenced using PacBio?
I say yeah. This is just the beginning. We do know that, like I said, HiFi sequencing can be sequenced for anything you can make a smart bell library out of. I would love to hear if any of those in attendance have thoughts about using PacBio for other viral vectors. Please reach out to us. Trey, I just wanted to also, you know, say that we know there are questions in the question box, and I know we will not get to all of them in the next 30 seconds. I believe that the go-to meeting will save these as a, like, we'll have to be able to contact the attendees. I know there are some questions there that are pretty technical for you, Don. It's regarding the natural filling. So we'll triage those later, and then probably send it back to Don, you, so that you, and I think there's a question for Terrence as well, that we can send it back to the person who proposed the question. Can you see the questions, panel, Trey?
I am not seeing the full panel, so I
Okay,
apologize for that.
then actually let me maybe since yeah, so we are at time, and I think it's we shouldn't assume that the person who asked can stay on. So
Yeah, we'll get directly
let's
to
just
that,
say we'll keep
Liz.
these questions there.
Mhm. Question, Liz, are you able to read that off, the question from the chat window?
I can't, but I'm saying we're at time, and I think it's better to not assume that the people who ask these questions can stay on, and that we will answer them. Like there's a way to record these GoToMeeting questions, and then we'll respond via email later.
All right, I think we have time for a couple more questions. However, I think these are important that I'm seeing. So maybe a question for Dawn. Really quickly, Dawn, you talked about using bacterial epigenome data. So the question is, how does incorporating bacterial epigenome data, specifically the 4MC and 6MA, help with characterizing AAV?
So when you've got your, if you think about it this way, for one, you want to confirm that you've created your blueprint essentially correctly. So obviously that involves cloning and Gibson and whatever else you're going to stitch everything back together again. For that, you essentially want to do kind of a sanity check where you're going to just sequence your plasmid. And that's essentially to show that what I built is the way I built it, and it looks exactly, the way it should look. So for that, then you're like, oh, okay, good going. Now it's ready to go over to the bioreactors and for us to then slap it together and put it in there and wave the magic wand. And then out of that, you have the cells essentially spitting out capsids that are full. And then that is where it comes into, you got your other methods for confirming, do we have full package, fully encapsulated genomes, or some of them partial? So then obviously for us, then you make those decisions on, do I need to go back to the drawing board and then use this method to redesign, or does everything look good? And, is what you're actually getting out of it mostly fully, full length genomes, in which case you really don't need to worry about them being broken. You just need to, do that last step where you are hybridizing your genomes, positive and negative strands, and you sequence them, and, thumbs up.
Great, thank you. And then maybe one final question, and this question is going to be for you, Liz. So I think you talked about in your presentation that AAV vectors can vary greatly depending on its design. So can you tell us a little bit about how PacBio can help researchers with implementing the workflow for AAV to either validate or characterize a specific design?
Well, I think that's what Don is showing with the way that he's and also the publications that we're seeing, right? And the report that I'm showing that we can generate, so we can show whether there's truncation events, where the mutation hotspots are, and whether, I think one thing that wasn't clear is that actually when people think they're producing single strand AAV, it's been shown that truncated products, versions of that can actually form a self-complementary structure, which might not be ideal as well.
All right, so again, as Liz mentioned, we are at time. We wanted to get those final questions through. So finally, I'd like to thank Terrence, Donald, and Don, and Liz for their time today and for sharing their expertise and experience. So in closing, I wanted to briefly mention a couple of things. First, in the next few days, we will be sending an email with a link to the recording should you want to go back and listen to any part of this webinar again. Second, you can also stay up to date on upcoming webinars and events by visiting and bookmarking pacb.com/events. And then finally, immediately following this webinar, you'll also receive a very brief questionnaire. Please do take the 2 minutes that's required to fill it out. It helps us to understand your needs and plan for future webinars. So with that, thanks again for joining us today. I hope you'll all join us again for a future webinar. And with that, take care and have a wonderful day. Thank you very much.
Thank you.
Thanks, Ryan.
Thank you.